Hello everyone. Today we'll discuss the topic oogenesis. Oogenesis is the formation of ova during the reproductive cycle, and this oogenesis process also involves the multiplication phase and the meiosis stage. This Here, the primary oocyte is formed during the first meiotic division and that give rise to the secondary oocyte and the first polar body. This first polar body it give rise to the two polar bodies, and the secondary oocyte after the fertilization at the time of fertilization releases the another polar body, that is a third polar body, and the ovum proper. That means at the end of the oogenesis, there is a formation of the three polar bodies and a single egg. In spermatogenesis, there from the uh, primary spermatocyte, there is a formation of the four sperm. Instead, in oogenesis, there is a formation of the three polar bodies and only single egg during the maturation. The oogenesis process it involves the two important phases. First one is a pre-meiotic phase, or that we can call it as a proliferative phase, and the second phase is a meiotic phase that includes growth of oocytes, vitellogenesis, maturation, and egg membrane deposition. The first phase is the proliferative phase. The oocyte arises from the primordial germ cells that are formed very early in the development and multiply by mitosis to form the population of oogonia, from which the oocytes will develop or arise. The proliferative phase is the period of oogonial proliferation within the ovary during the larval or fetal life. In mammals and birds, the primordial germ cells proliferate in a limited number of oogonia, and it is completed once in a life of an organism long before the sexual maturity. In fish and amphibian, proliferative phase is a seasonal throughout the adult life of the organism. All oogonia are not matured into functional eggs since they do not survive into the adult. Surviving oogonia usually begin meiosis in larval stage and fetal life, and these becomes a primary oocytes. This marks the transition stage between the pre-meiotic phase and the meiotic phase. The egg development in vertebrate is a very wasteful process, and in the early life that 1700 primary germ cells migrate to invade the undistinguished female gonad or ovary at 1 month of the fetal development. They proliferate in 2 months of gestation, and now at the 2 months the gonad possesses the 6 lakhs of the oogonia, and by fifth month there are 7 millions of oogonia. Mitosis stops at this stage, and many of the oogonia enter the first meiotic prophase and becomes a oocyte. Many oogonia undergo degeneration, and so that at the time of the birth only 2 million oocytes remain. It continues to degenerate, and at the puberty or sexual maturity only 3 lakhs of the oocytes remain. During each month, several oocytes are stimulated to mature, but only one completes the meiosis and it gets ovulates or released from the ovary and other degenerates. Throughout the entire reproductive life of women, only 400 to 500 ova will have been mm, ovulated and other will degenerate. At the time of the menopause, no oocytes can be detected in the ovary of the woman. Then the next phase comes, that is a meiotic phase. Before oogonia enter the meiosis and become primary oocyte, their chromosomes replicate and amount of DNA it gets doubles and prophase 1 is prolonged. Arrested prophase is the most peculiar feature of the oocyte meiosis. During meiotic prophase, oocyte grows and acquires a developmental information. The meiotic phase can be divided into first phase, that is a growth phase, which involves the accumulation of nutrients necessary for the embryonic development and the genetic programming for the development. Example in Xenopus, 1 lakh times more DNA polymerase enzymes than histone proteins, mitochondria, and ribosomes as compared to the somatic cells or the body cells. Such holding is required for rapid cell division during the cleavage. The oocyte grows to an enormous size to become the largest cell of the animal body, and this growth occurs due to increase in the reserve material such as yolk. Rate of growth of primary oocyte may be slower or higher as per the organism. Second one is the growth of the nuclear substance. During the meiotic phase, the Lambrus chromosomes are found in the oocytes, which are responsible for the bumper synthesis of the mRNA from the oocyte genome. The mRNA synthesized during oogenesis are of 3 types. That is, first one is immediately translating mRNAs. Then the second one is what? The heat shocked mRNAs, and the third one is masked mRNAs. Immediately translating mRNAs immediately produce the ribosomal proteins, histone and enzymes, etc. Masked mRNA, which is activated only after fertilization, and the heat shocked mRNA induced to translate into the proteins at high temperature. The synthesis and storage of ribosomes occur, which sustains the embryo during its development till it begins to synthesize its own ribosomes after hatching. A Golgi apparatus present in the oocyte also performs the variety of functions. They perform the secretory, storage, and the transport functions. The stacked lamellae vesicle around centrosome starts the vitellogenesis and helps in the synthesis of yolk. The Golgi apparatus is also involved in the synthesis of cortical granules, which in later stages move to periphery and get arranged as a layer close to plasma membrane of the oocyte, which forms the cortical membrane or the fertilization membrane just after the fertilization. Then the next process comes, that is a vitellogenesis or synthesis and deposition of yolk during the development of egg. Yolk accumulates during the growth phase, accounts for rapid increase in size of maturing egg. Chemically, it composed of proteins, phospholipid, and neutral fats. It may be called protein yolk when more proteins than the lipid, or Fatty yolk when it has more fat content than the proteins. Yolk occurs in two forms. One is a granular yolk and another one is a yolk platelets. The granular egg found in egg of invertebrates and lower chordates such as amphioxus or tunicates, which remain evenly distributed in the cytoplasm of the oocyte. Whereas the another form, that is a yolk platelet, it is found in most of the vertebrate groups like the cyclostomes, fishes. In fishes also the lungfishes, dipnoids and the elasmobranch and the teleost, then amphibians, reptiles and birds. The yolk occurs in the form of large granules, and these large granules are called as a yolk platelets. Cytoplasm is densely packed with them. In amphibian oocytes, the two main proteinaceous substances are found. One is a phosphatidyl and another one is a lipovitellin. In the yolk platelets, two molecules of phosphatidyl are associated with one molecule of lipovitellin in a structural unit. Then the functions of ovarian tissues during the growth of the oocyte. In species lacking the Lambrus stage, oocyte receive information and nutrient reserves from another type of the surrounding cells. These cells are called as accessory cells such as nerve cells and the follicle cells. The metabolism of both the type of these cells is coupled to oocyte growth. The intimacy is so great that direct cytoplasmic connection between nerve cell and follicular cells and the growing oocyte is there. The principal role of these cells is to supply oocyte nutrient reserves during its growth, act as a selective barrier between vascular system of oocyte, transporting essential molecules, yolk precursor into the oocyte cytoplasm, and synthesize the accessory egg membrane. The first one is nerve cell. These are found only in invertebrates such as coelentrates, annelids, mollusks, and insects. They are derived from the same oogonium that give rise to the oocytes. During oogonial divisions in the ovary, at the same points, a differential division separates cells to destined to become oocyte from their sisters which develop into the nerve cells. For example, in insect water beetle Dytiscus, a large fulgen positive mass DNA in an oogonial nucleus is segregated into only one of the two cells following an oogonial division, so that only one cell out of 16 cells retains the extra DNA, which become destined to form a oocyte, whereas other without Fulgen positive body becomes the nerve cells. And these nerve cells, these that is the from the cluster of the 16 cells, these 15 become the nerve cell and the one become a oogonia. In Dytiscus, this extra DNA represents the mass of the ribosomal RNA genes, a mechanism of gene amplification in amphibian oocyte. Oocyte and nerve cell develop from the same cell within a germarium, a constricted chamber in the ovariole. A single stem cell undergoes a differential division. One daughter cell remains as a stem cell while other is called as a cystoblast or oogonia, which completes the four mitotic division to form the cluster of 16 cystocytes, one of which become a oocyte and rest of the 15 becomes a nerve cell. The follicle cells surrounding all the cystocytes. Nerve cells connective to oocyte during direct cytoplasmic bridges or ring canal. The cell retaining the maximum number of cytoplasmic bridges is a future oocyte. The presumptive oocyte usually appears at the first division, while all subsequent division it produces the nurse cells. Then the next cell, that is a follicular cells. These are present in vertebrates and other groups. In vertebrate, these cells are derived from the germinal epithelium and become organized as a single layer of epithelial cell during the developing oocyte, as in the human ovum. Though they synthesize some substances for oocyte storage, they act as a selective barrier, that is the materials from bloodstream to oocyte have to pass through the membrane of the follicular cells and determines what material to be allowed through. It facilitated this transfer of process. There are thousands of cytoplasmic processes which are called as a microvilli reaching out from the surface of both the oocyte and the follicle cells. These microvilli interdigitate to some extent, thereby greatly increasing the surface area over which contacts and thus exchange can occur. In contrast with nurse cell, follicular cells do not arise from the oogonia and are not in direct communication with oocyte and generally transport rather than synthesize the oocyte bound material. This is the human ovum or the young mammalian ovum surrounded by the follicular cells. And these follicular cells having the or these ovum having the processes of these are the follicular cells which interdigitate for the transportation. And these are the follicular cells around the ovum. In mammalian oocyte of the 100 micrometer in diameter develops within a connective tissue called the follicle. As oocyte proceeds toward the maturation, it becomes larger and filled with a liquid that is called as a liquor folliculi, and the oocyte becomes surrounded by more and more follicular cells. At the mature stage, follicle is called as a Graafian follicle and having a eccentric fluid filled cavity that is called as the antrum, having the antral fluid. A thick gelatinous membrane, that is a zona pellucida, separates the oocyte from the layer of the follicular cells. The corona radiata is the layer which is several cell thick, and the follicle cells also surround the antrum and are responsible for secretion of the antral. Then the last process in the oogenesis is the formation of the egg membrane. In most of the animals, except coelenterate and sponges, oocyte maturation is not completed until the additional structure called as a egg membranes or egg envelopes, which are produced outside the plasma membrane of the egg. The first one is a primary egg membrane. The egg membranes are formed in the ovary and are laid between the egg plasma membrane and the follicular cells. These are formed either from ovum or follicular cell and sometimes by cooperative efforts of both. Primary egg membranes are of following types. First one is a vitelline membrane. It is this is not a membrane but a non-cellular transparent layer of mucoprotein forming an extracellular matrix which is both fibrous and amorphous. It is often much thicker and stronger than plasma membrane of egg and has a tensile and elastic properties. It is given different names in different animals. Example, in fish it is called chorion and in reptiles and mammals it is called as a zona pellucida. It is usually remain intact through several stages of cleavage where it helps to hold the dividing cells together. Then the next layer, that is the zona pellucida, the primary envelope of the shark. Some bony fishes, some amphibians, and some reptiles have striated appearance and is called as a zona radiata. which it represents the graded microvilli of the growing oocyte. Then in echinoderms, the jelly envelope is there, and many other eggs of marine invertebrates also have such type of the envelope. The primary egg envelope is much thicker structure of the jelly. All these primary egg membranes are attached with the surface of oocyte, but at later stage, a space filled with a fluid may appear between the egg plasma membrane and primary egg membrane, and this is called as the perivitelline membrane. Then in some eggs, there is a small opening, and that small opening is called as a micropyle, which is found in the primary egg membrane. Sperm usually makes its entry inside the egg during fertilization through the micropyle. Example, it is present in Pisces at animal pole and at vegetal pole in the mollusks. Then the normal procedure or the process of oogenesis in human beings. As earlier discussed, this is the primary oocyte which is formed, and this that is, which is formed. This primary oocyte it divides in the meiosis one, and it gives rise to the what the secondary oocyte, which will be the and the what it gives rise to the first polar body. In the second division, secondary oocyte giving rise to an ootid or the ooham proper and releases the one polar body. And this first polar body again divides to give rise to the two polar bodies. The second division of this oocyte does not occur unless fertilization uh, by the sperm. The process of human oogenesis at the start of menstrual cycle, 12 to 20 primary follicle begin to develop under the influence of the elevated follicle stimulating hormone to form the secondary follicle. By the around 9 day of the cycle, only one healthy follicle is remaining, with rest having undergoing the atresia. The remaining follicle is called as a dominant follicle, which is responsible for producing the large amount of the estradiol or estrogen during later follicular phase. The estradiol production depends on the cooperation between the theca and the granulosa cells. On the 15th day of the cycle, the LH surge, that is secretion from the that pituitary, which is triggered by the passive feedback of the estradiol, and this causes the secondary follicle to turn into the tertiary follicle, which ovulates some 24 to 36 hours later. An important event in the tertiary follicle is that primary oocyte completes the first meiotic division with the formation of polar body and the secondary oocyte. The empty follicle then forms the corpus luteum, which is a temporary endocrine gland. This is a ovary from where the primary oocyte is formed, which give rise to the second. This is a primary oocyte and having the what the zona pellucida is covering, and then this is a maturing follicle, and after the what that is a first meiotic division, and it having the antrum and the what that is the first polar body is released. So this is a process of the what how the oogenesis occurs. This is the oogonium or the stem cell after the mitosis. It forms a what the stem cells. It reserve cells and it give rise to the what the primary oocyte, and this primary oocyte this forms the give rise to the secondary oocyte and the first polar body. This secondary oocyte after fertilization it give rise to the another second polar body, and this is a ovum proper, and this first polar body it again divides by the meiosis 2 to give rise to tubes, and then therefore at the end of the what at the end of the oogenesis there is a formation of the three polar body and the single ovum. This is the primary follicle or oocyte. This is the primordial follicle. Then it develops into the primary follicle. Then secondary follicle or growing follicle. Then vesicular or the antral follicle. And at the 14th day of the cycle, what happens? There is a release of the ovum proper from the uh, Graphian follicle into the abdominal cavity for the fertilization.